Welcome one and all to the very first of my review videos or review lectures or whatever you like to call them. But it's for the class Biology 110 for the fall 2021 class. And this first one goes over lab number 2, which is protein electrophoresis and ionization. I hope I'm saying those correctly because I have no idea what I'm doing because I'm also a student in this class. But I decided to make this just because I think teaching is probably one of the best ways of learning. So basically, in this lab, there was an introduction and it said that there was this thing that are called amino acids, and they're linked by peptide bonds. And we're going to learn this later, but we're going to find out that amino acids are actually the building blocks for proteins. But he also said that, Professor Humphrey said that in lecture. So electrophoretic separation depends, that is what we're using to separate out proteins in this lab, and those proteins can be represented using dyes. And we're seeing how proteins in different pH solutions will move across a gelled surface differently. So that's what we were doing. We were creating the gelled surface by stirring it in different pHs, and then we were putting proteins on top of that and seeing which way they moved depending on the concentration of the H+ ions within each solution. And this was determined we saw by the R groups, which were either carboxylic acids, as shown here, or amino groups with NH3+. Now, the funny thing about this, and I misunderstood this at the start of the lab, but it turns out that the O-, when the carboxylic groups, when they're in high pH, they'll lose a proton and form water. They'll form H2O at high pH because there are an excess of there is a deficit in the concentration of H+ ions, and then in low pHs they will be neutral because they will have their OH ions at the end of their carboxylic acid group, and they will bind to them, and the protein will be neutral. And the opposite is true with the NH+ group, NH3+ group, because the NH3+ group gets positively charged at low pHs because the nitrogen atom will actually bond to an extra hydrogen atom and form 4 bonds and become positively charged, and at high pH it'll be neutral. And I drew some graphs because it's honestly really hard to visualize how these atoms charge in low and high pHs without graphs, so that's what they look like here. Moving on to focus questions 1 through 5. The first focus question is, what is the major property of proteins that influences their migration in an electric field? Which type of amino acid R groups is primarily responsible for this property? So basically what this question is asking is, why do proteins move in an electric field? That's the first question. And that one's pretty obvious. That one is because their R groups get charged based on the pH. So they're trying to say, how do they get positively or negatively charged? It's because they're gaining this proton or losing this proton, and that charges that ionizes these proteins as a whole. So that's why it's electrophoretic and ionization. And then the second part of this question, which type of amino acid R groups is primarily responsible for this property? Well, it would be the carboxylic and the nitrogen, what are they called? They're the amino groups. So, yeah. Okay, moving on to question number 2. What distinguishes aspartic acid and glutamic acid from lysine, arginine, and histidine? Well, first of all, you can see by the nomenclature that aspartic and glutamic are acids. So that means that in water, they will lose a proton and become negatively charged. So that's one thing. Because lysine, arginine, and histidine all have these nitrate groups, while aspartic acid and glutamic acid have these carboxyl groups. And that was shown for us in the earlier diagrams. So that's a few things that distinguish them. What happens to the charge on a charged amino acid as pH changes? Well, as pH changes, the charges will either become more extreme or go to neutral depending on what they have inside the protein. It all depends on what the protein is composed of. How is it that different proteins can have different net charges? How is it that the net charge on a protein changes with pH? What is the isoelectric point of a protein? Okay, so let's break this down because it's 3 questions. How is it that different proteins can have different net charges? They can have different net charges because, again, of what they're composed of. If a protein has a bunch of aspartic acid and very little lysine, then that protein is going to tend to be much more negatively charged in high pHs and much less positively charged in low pHs. How is the net charge on a protein change with pH? Well, I think I answered that one in my last answer, which is, again, what it's composed of. Oh my gosh, take a shot every time I say what it's composed of. Anyways, what is the isoelectric point of a protein? An isoelectric point of a protein, this is a new question, is the point at which a protein has no net charge. So that is the pH at which a protein will have a neutral amount of positively and negatively charged ions in its structure. Okay, well that's all I'm going to do for now, because I'm lazy. Yeah, but I think that pretty much accurately sums up all of the critical points of the lab. If you have questions, I guess approach me outside. Catch me outside, catch these hands, and if you're a professor and you have a problem with me posting these videos, I will stop doing it and I'll take it down. I'm sorry.